Hi there, my name's Steve Yang, and I'm here to talk to you about my research project that I'm doing over the summer through the leadership program up at the Baker Institute in Dr. Vanderwall's lab. So who is Dr. Vanderwall? Well, Dr. Vanderwall is this. This is Dr. Vanderwall. She's smiling. So who is she though? I just showed you her picture, but more importantly, her research is primarily on cancer, looking for treatments for canine and feline mammary breast cancer. She's looking for novel drugs that can be used to fight off herpes infections, primarily equine herpes virus infections and feline herpes virus infections. And finally, she is doing research on stem cells, primarily mesenchymal stem cells, and looking at their therapeutic value in terms of wound healing, antibacterial properties, or my project this summer, which is on their antiviral properties against primarily EHV1. So far our lab has already shown that mesenchymal stem cells produce factors that have some sort of antiviral properties that inhibit the growth of EHV1 within certain epithelial cells that we have cultured, primarily equine fibroblast cells. So my project this summer is to identify the mechanism by which our mesenchymal stem cell exudate or conditioned medium does this. And my conditioned medium has actually already been lyophilized, which basically means it's been dry frozen or cryo desiccated, essentially, into a powder form that can be reconstituted by the veterinarian during times of stress for the horse to perhaps lower the infection that occurs from latency. When the horse gets stressed out, the virus reactivates. So what is EHV1? I mentioned the concept of latency, but that's only one part of it. EHV1 is an alpha herpes virus that primarily infects the respiratory epithelium of horses first through secretions and close contact. Afterwards, after causing rhinotracheitis or bronchitis, etc., rhinopneumonitis, it can undergo cell-associated viremia, which means it goes through the blood. And what owners are really concerned about is abortions that can occur in neurologic signs, primarily equine herpes myeloencephalopathy, or EHM, both of which, and they harp on this a lot in vet school, are caused by a sort of vasculitis. So it's more of a vascular problem, and the virus itself does not infect the nervous system. Pretty cool. So my project, aside from identifying the mechanism through which the mesenchymal stem cells, as I mentioned, inhibits equine herpes virus infection, is also to see if these equine corneal epithelial cells that we have collected, or someone's collected from horses, are a good model for infection as well, since the cornea can be infected naturally in horses with herpes virus. It's rare. The cornea is usually affected in like canine herpes virus or something, but we want to see if that's possible as well. The experiments we've done so far include, but not limited to, just some of the experiments I've done include PCR on various genes that may have been upregulated during infection to see if the mesenchymal stem cells can decrease the upregulation of those genes or increase upregulation of certain genes that I found during a literature review. Staining the actin cytoskeleton of epithelial cells to see if the actin changes during infection and to see if my mesenchymal stem cell conditioned medium can inhibit the changes in the actin cytoskeleton, since, as we all know, the cytoskeleton of a cell is very important during virus replication. So at this point, for the actin cytoskeleton, I've only stained them and looked at them. I haven't quantified anything yet. But I hear quantification takes a long time. And then finally, I've started infecting my equine corneal epithelial cells to see if they are a good model. And so far, it doesn't seem like they are. But we'll see. We'll get more results. I'll let you guys know later on. Did I miss anything? Nope, I didn't miss anything, I think. I forgot to show you guys the lab specialist that I'm working with. She's super smart. She knows all the laboratory techniques. And I'm very happy to be working with her because she's super nice. Her name is Rebecca Harmon. I have a picture of her as well. This is her. I'm sorry it's in black and white. Which is pretty cool. All right, thank you guys for listening. Let me know if you have any questions. But I won't be able to answer them because I'm not there. All right, see you guys. You can email me questions if you want. Okay. Hi, John.